Okay. Thanks very much, Mario. Good morning, everyone. As Mario said, I'm going to be talking about genomics this morning. So just for those of you who I haven't mentioned, an engagement team leader at ABRI, and I'm based in Armidale in New South Wales in Australia. I have met a number of people in Southern Africa. I've been across to South Africa to attend the Stockman School twice now. So always a great event, and really enjoy getting to talk to the South African breeders and experiential production systems. So today I'm going to be talking about what is genomics, what is single-step breed plan, what are the benefits of single-step. Then we're going to look at some case studies to see what impact single-step breed plan has had for a number of different breeds. And then finally finish up with some considerations. So talking about performance recording and considering which animals you might like to genotype. So you may have heard the word genome. When we talk about genome, what we're talking about is the genetic material of an organism or an organism's DNA. So we've got our organism, in this case, our bull. He's made up of cells. Inside the cells are chromosomes, and those chromosomes contain the DNA. So that's all of the animal's genetic material is contained in his cells. And that's what we're talking about when we talk about the genome. So recently, in recent years, DNA testing in beef cattle has become more widespread. And so we're at the stage now where producers can take a sample from an animal, send it into a commercial genotyping lab and get a DNA test done. Now, there's three main sample types that we're seeing being used by producers around the world. They each have pros and cons. The one we've seen become more and more common in recent years is a tissue sample, which is in this first picture here. You can see the sample tubes and then the applicated gun. That is often taken in conjunction with calf tagging, and it involves taking an earplug sample from the calf's ear. The good thing about TSUs and tissue samples is that you can collect them from birth, but it does require you to have a specialised applicator gun and also to purchase your TSU tubes. And the other downside is that they're not suitable for long-term home storage. So they're not suitable at this stage as a backup option for you to store samples of individual animals at home and farm. The second type that we see used commonly is hair samples. Now, though that involves pulling approximately 40 hairs from the tail switch, it's really important that if you are taking hair samples that you pull them and you don't cut them, because the DNA is contained in the hair root. And if you cut the hair off, you're not going to get the root of the hair. So really important that they're pulled and the bulbs and the roots of the hairs are included in your sample. The nice thing about this as a sample type is it is really quite stable at room temperature, so it's suitable for long-term storage. If you are storing hair samples at home as backups for your animals, we do recommend that you store them in a paper envelope rather than a plastic envelope. If you store them long-term in plastic, they can sweat and go moldy. The downside, though, is that they're not recommended for collection from calves that are younger than 60 days of age. So they're not typically samples you can take at birth. And then our third option that we see being used to collect samples for DNA testing are semen straws. That's really useful when you've got an animal that is physically unavailable, so it might be located overseas or it may be deceased. So you can use semen straws to get a sample of that animal, obviously only applicable to mature bulls. And so once you've collected your DNA samples, there's really four major applications that you may be doing this for. The first is for parent verification, to identify who the animal's parents are. That can be either confirming the sire, confirming the dam, or both. Also to help you manage genetic conditions. We see a range of genetic conditions in different breeds. Some of those have genetic condition tests available, and so people will take samples to see whether or not animals are carrying genetic conditions. Can also be used to help you manage qualitative traits, so things like coat colour, red and black, for example, or horn or pole status. And then lastly, it's used in genetic evaluation. And today we're going to focus on the application in single-step pre-plan. We know that in reality, most people are not doing a DNA test for one of those reasons. It's often a DNA test done for a combination of the reasons that I've just mentioned. So, great. I've talked a little bit about single-step pre-plan. Currently, ABRI has 19 single-step pre-plan analyses that we conduct for breed societies and breeder groups around the world. The first of these was implemented back in 2017, so we're coming up to having a couple of single-step evaluations that have been running for 8 years or more. At this stage, we have over 1.15 million genotypes that are being analysed across all of those single-step breed plan evaluations. And there are 3 single-step breed plan analyses that are including breeds from across Southern Africa. So the first of those is our Hereford single-step breed plan evaluation that combines data from Herefords Australia, New Zealand Hereford Association, and also the Hereford Cattle Breeders Society of Namibia. That was first implemented back in October 2017. Brahman, so combining data from the Brahman Cattle Breeders Society of Namibia, South Africa, and also Brahman breeders recording the Zimbabwe herd book. That was implemented in November 2023. And then lastly, Limousin. This is essentially a Southern Hemisphere analysis. It includes Australian Limousin breeders, which also includes breeders from New Zealand, and then the Limousin Cattle Breeders Society of Namibia and South Africa. And like Brahman, that was also implemented back in November 2023. And we're always developing more single-step breed plan analyses. We have one that is due to come out this month for a breed in the UK. So certainly seeing more and more breeds looking to incorporate genomic technology into their breed plan analysis. So what is single-step breed plan? It's about incorporating the pedigree information, the performance information, so the phenotypes or the performance data that you collect on farm, and also the genomic DNA information into the calculation of the EBVs. So in conventional breed plan, we include the pedigree and the performance information. And in single-step breed plan, we still include the pedigree, we still include the performance information, but we include DNA or genomic information as an additional source of information for breed plan to use. In order for us to switch on single-step breed plan for a breed, we require a reference population. So that is animals that have both phenotypes or performance data, so weights, scan, fertility information, as well as genomics or DNA samples. And it's really important when we're building that reference population that that reference is representative of the whole population. So we want to make sure it's capturing all of the variation within that breed. And that can be particularly challenging for breeds that are composite or have diverse backgrounds. So, for example, if we look at this example here on the left-hand side of the screen, this is a Brangus. I'm aware in South Africa, Southern Africa, there, Brangus are typically red. I couldn't find a photo of a red animal, but a breed that has a background of Brahman and Angus, typically, so composite background. And also diverse backgrounds. So, for example, I've certainly mostly seen traditional Simmentala when I've been across in Southern Africa, but here in Australia, Simmentala would be an example of a breed with a diverse background. We have both the traditionals, like you see here, and then also some black animals and some red animals. So, different backgrounds, diversity within that breed. So, those sorts of situations can be a little more challenging, but there's certainly we can still do single-step evaluations for them. It's just that we have to be very particular about what that reference population looks like as we're doing it for breeds that are composite and diverse. Okay, so at its basic level, what we're really doing with genetics is describing relationships between animals. We know, so this here is a photo of my brother and I and our parents. And my brother and I have both got half of our DNA from our mother and half of our DNA from our father. So they've passed 50% of their DNA onto each of us. But each sibling does not get exactly the same DNA from mum or DNA from dad unless you're an identical twin. And I think that intrinsically makes sense if you have siblings and you think about yourself and your siblings, or if you've got kids and you think about your kids, you can see family similarities, but we're not identical, we're not clones. And so we're getting different bits of our mother's DNA and different bits of our father's DNA. They mix together, and then we've got our own unique genetics that we've received from both of our parents. The same is true for cattle. So here I've got an example of a sire and a dam, and they had 10 full sibling ET calves, including the 3 bulls below. And so if you look at these animals, you can see that yes, there are some family similarities, but they're not identical animals. So on average, they're sharing 50% of their DNA. So single-step breed plan is all about more accurately describing the relationships at the DNA level between individual animals. So if we think about our conventional breed plan, where we don't include genomic information, the analysis assumes that we get 25% of our genes from each grandparent. So you can see that here, here's the sire. He's got a blue chromosome that he's got from his own sire and a red chromosome that he's got from his own dam. And you can see, if we look at his offspring, it's this offspring has got 25% blue and 25% red. So it's got exactly equal amounts from the paternal grandsire and the paternal granddam. And the same if we look on the female line, you can see the dam has inherited a pink chromosome from her sire and a yellow chromosome from her dam. And if we look at this offspring, you can see the assumption is that it's got exactly 25% of the pink chromosome and 25% of the yellow chromosome. So everything's being passed on equally from each grandparent. That's what we assume under conventional breed plan. But we know that's not the case. We don't all get 25% from each individual grandparent. We have a biological process that happens, the DNA gets mixed up, and so you get variable levels of genes from each grandparent. And you can see that in the second example here. So you can see this particular animal, it's still got half of its DNA from its sire, but you'll see it's got more of its paternal granddam's DNA than it has of its paternal grandsire. And if we look at the dam line, same thing, it's still got half of that DNA from its mother, but it's got more from the maternal grandsire and less from the maternal granddam. And so that's what we're describing in single-step breed plan. We're getting a feel for the relationship at the DNA level that an individual animal has with its relatives and what percentage of the DNA they share. So what does that tell us? Well, there's 2 main benefits that we see from incorporating DNA information into the breed plan analysis. Firstly, we can see EDVs generated for animals that do not have performance data. Now, there's a whole range of reasons why an animal might not have performance data. There may be animals from non-breed plan herds, so they're not being performance recorded. They may be young animals that aren't old enough to be measured for a trait. So if we think about the breed plan traits that you will have gone through in the previous webinars, most of those traits are measured on an animal from when it's born through to when it's rising 2-year-old. So those 600-day weights, ultrasound scans, for example, they're typically taken on animals that are nearly around 2 years of age. So a young calf, like this one we see on the picture here, is not old enough to be recorded for 400-day weight or 600-day weight. So it might not have trait data for those yet because it's just not old enough. We also have a range of traits which are hard or expensive to measure. So net feed intake and methane are good examples. They are typically requiring animals to be in feedlots on feed for a number of days. And so They are more expensive to measure than something like a live weight, which we record by taking an animal over a scale at a single point. So not all animals will be recorded for those sorts of traits. Certainly in Australia, we see a lot of those traits recorded in research programs, not so much on farm, although I know there's a number of producers across Southern Africa that are doing a good job of collecting net feed intake on farm. We also have traits that are only measurable in one sex. So a good example there are our female fertility traits and also mature cow weight. Carcass traits taken in the abattoir are a good example of that. And obviously for those of you that are breeding stud animals, that's not something that you'll be able to collect a lot of. The second thing that single-step breed plan does for us is we can get more accurate EBVs generated for animals with limited performance information. So these are typically our young animals that have their own performance data but no progeny, and that's typically the sale bulls. So if we've got yearlings or rising 2-year-olds that are available for sale, they'll be old enough to have been performance recorded for their traits, but they won't yet have progeny on the ground that can be performance recorded. And also animals that are in small or single animal contemporary groups. And so that will include often animals like ET calves, where there might only have been a small ET program. Twins are another good example within the breed plan analysis. We only compare twins with other twins. We don't compare them directly with calves that are born single. So most herds won't have a large number of twin calves each year, and so they end up in small single animal contemporary groups. And also those breeders that have smaller herds that may only have a handful of animals, they often will have smaller contemporary groups just because of the number of animals that they're running. Okay, so having more information, having EBVs available, having more accurate EBVs allows breeders to make more informed selection decisions at younger ages, and that will shorten the generation interval and give you the potential to increase your rate of genetic improvement. Have we had any questions come in at this stage, Azan or Mario?
Not yet.
Okay. I've got a couple of question breaks throughout the presentation, so if anyone's got questions, please type them in the chat, and I'm sure Azan will let me know when they appear in the chat. Okay, so that's the theory. Is single-step breed plan really delivering benefits for beef producers? So to answer that question, I've got a couple of case studies that I'd like to take you through. The first is Australian Brahman, and it's a really good example of EBVs being generated for animals that do not have performance data. So they were the first breed to introduce single-step breed plan back in April 2017. And historically, within Australia, we've got two types of cattle. We've got our Southern Australia production systems, which are typically Bos Taurus, so down in the southern states below where my mouse is on the screen. They're typically more intensive production systems, smaller blocks, intensively managed, and therefore have good opportunity to record for breed plan. Whereas in the north, across Queensland, Northern Territory, and the top half of WA or Western Australia, they're more typically Bos Indicus breeds or they're composites. Those production systems tend to be more extensive. They're large parcels of land, and because they're extensive, it has been more challenging for beef producers on those properties to collect performance records. And so historically, we've seen that perhaps our Bos Indicus breeds have not utilised genetic evaluation to the extent that their Bos Taurus counterparts have, simply because of the challenges involved in collecting performance data in a more extensive production system. And Australian Brahman have a large multi-vendor sale in October every year, which is called Brahman Week. And we, for anyone who would like to read this case study in more detail, we do have a paper that's just come out 7 years on what a single step breed plan meant for the Australian Brahman breed. That was presented in June in New Zealand. So if anyone would like to read that in more detail, please let me know and I can provide you with a copy. So at Brahman Week back in 2016, before we introduced single step breed plan for the breed, you can see that less than 20% of the nearly 900 lots had EBVs for a range of traits. So you can see here, percentage of sow animals with EBVs, birth weight, 200-day weight, 400-day weight, 600-day weight, scrotal size, days to calving, eye muscle area, and rump fat. Very few animals in the sow catalogue had EBVs available. So for the majority of animals, we didn't have a good feel for what their genetic merit was. Last year, it was over 90% of the sale animals that had EBVs available. So that's been a huge success. It's allowed vendors that are allowed people that are looking to buy animals to have a good feel of the genetic merit of those animals. And so that has largely been driven by genotyping. You can see here that it hasn't happened instantaneously. There has been from 2016, 2017 was when single step was introduced through to 2024. You can see the number of animals with EBVs tracking up. That's just an example for days to calving, but you see a similar pattern in the 600-day weight. And so, as I said, driven largely by the widespread adoption of genotyping within the breed, including by those that don't performance record. The vast majority of animals in this catalogue last year with EBVs, that was driven by the fact that they have been able to take a DNA sample, get a genomic record, and have EBVs that are being driven by the genotypes, which is really fantastic news in terms of understanding the genetic merit of the sale animals. So as I said, EBVs have been generated for animals that have been genotyped but don't have performance. And it is really worth pointing out that that's really only been possible because Australian Brahman have a large-scale reference population. They have lots of animals with genotypes and lots of animals with phenotypes that can help drive that single-step breed plan evaluation. And that's because the breed has done a fantastic job of being involved in research projects that have collected phenotypes and genotypes on a range of traits. They've had a dedicated genomics project for a large number of years. They've been involved in that, which has focused on collecting female fertility in Brahman and several other tropical breeds. And they've also got a northern steer bin project where they're collecting the carcass data in the abattoir on the genomics projects. The heifers out of the genomics project, their brothers are going into that northern steer project and getting all of the carcass data. So I think that really illustrates the importance of phenotypes. I certainly don't want anyone to get the impression that you can just genotype and not worry about phenotyping. So I'll talk more on that later. But it is a lovely example of how we can take this technology into a region that's a bit more extensive, where we haven't had large uptake of breed plan EBVs on sale animals, and genomics and single-step breed plan can be used to get a feel for the genetic merit of animals that previously we didn't know anything about their genetics, and people that purchased them were really taking a stab in the dark as to what they were getting in terms of genetic merit. Our second case study is Hereford single-step breed plan. And so this is really showing the benefit of more accurate EBVs generated for animals with limited performance information. Hereford single-step breed plan was first introduced in 2017. It combines data from breed societies in Australia, New Zealand, and Namibia. At the time of this study, which admittedly was back in 2019, so only 2 years into them having introduced single-step, they had a reference population of around 10,000 animals. They have a significantly larger reference population than that now. And what we did was we wanted to have a look at what effect genomics was having on EBVs. So we did 2 runs. We did the first run where we included the DNA information. So we have pedigree, performance, and DNA information our standard single step run. And then we did exactly the same analysis, but we excluded the DNA information. So more of a conventional replan analysis. And it's important to note that we used exactly the same data set and just took information out. We didn't go month 1 and then month 2 for those different analyses, because things would then change due to additional data coming in. They were identical runs, just with and without genomics. Again, if anyone would like a paper, it is available and I can provide it. So what did we see when we did these 2 runs? We saw that average EBVs have not changed. If you look down at the traits and you look at the EBV average for conventional replan, so when we didn't have DNA information, and when at single step replan, when we did have DNA information, you can see that on the whole, the averages have remained the same. So that's good. That's what we expected. On average, if you look at the average EBV within the analysis, no change. But what we do see is that EBVs for an individual have changed. So what we have along the x-axis here is the replan EBV where we don't have genomics included, and then the replan EBV on the y-axis where we do have genomics included. And so you can see the line, if there was no change, animals will be right along that line. But you can see that we've got animals that have increased their EBV value when genomics or DNA information was included, and also animals that have decreased. Now, these are young animals, so we see quite a bit of movement in their individual EBVs because they were lower EBV accuracy than, for example, the sires. So in orange, we have the sires. They had EBV accuracies of 75% or higher. And you can see that we've still got some movement. Some their EBV has gone up, some their EBV has gone down. But the magnitude of the movement is not so great as what we see for those young animals that had lower EBV accuracies. So we do see movement, but certainly don't see as much movement for high accuracy sires. The second thing we saw is that variation in EBVs have increased. So we see more spread. And so what we've got here is the 200-day weight EBV values for some animals when we don't have genomics, and then when we do include genomics. And you can see we've got more variation at both ends. Now that's really valuable to beef producers and breeders, because it allows us to better identify animals at the extremes. We can better identify animals at the top end, which are typically the ones we want to use for our breeding programs, and also at the bottom end, which are typically the ones we want to cull from our breeding programs. So we're seeing greater definition of animal and greater spread for animals that are up here or down here. The third thing we saw was that EBV accuracy has changed. So along the x-axis here, we have the EBV accuracy in conventional replan where we have no genomics, and then the EBV accuracy when we included the DNA information. And so what you'll note is that for these animals that had a low starting EBV accuracy before we added genomics, we see quite an increase in their EBV accuracy when we include the genomic information. Whereas up here, for animals that had a high starting EBV accuracy already, they were 75, 80% accuracy or higher, we don't see so much increase in their EBV accuracy. So that's a pattern we see across different traits. This was 200-day weight. If we look at days to calving, we see a similar thing. Those that started with a lower EBV accuracy tend to have higher increases in EBV accuracy than those that had higher EBV accuracies to start with. So what we notice is that change in EBV accuracy is not uniform across animals. Those that started with lower EBV accuracies tend to have a bigger increase in EBV accuracy when we include genomic information than those that started with high accuracy EBVs. And that pattern is consistent across traits. The low accuracy animals usually have a much higher magnitude rise in EBV accuracy than those that were already high. But the average increase, or the jump, the magnitude that we see the EBV accuracy increased by, is not uniform across traits. And we saw that when we looked at 200-day weight and days to calving. We saw a much bigger jump in EBV accuracy when we included genomics for 200-day weight than we did for days to calving. And so that is not just because of the heritability of the trait or how much variation is explained by the genetics, but also by the number of animals in the reference population for the trait. And at the time that we did this study, there were certainly more animals that had a genotype and a 200-day weight than had a genotype and a female fertility or days to calving record. Now, as I said, that study was back in 2019. I certainly know that the reference population for the breed has grown substantially in that time. So I expect we'd see an even bigger impact of genomics coming through now in terms of lifting the EBV accuracy. But certainly at the time, that was the sorts of rates we were seeing. So I guess one really important message is that not only is improvement in EBV accuracy not uniform across traits, but it's also not uniform across breeds. And so this is a recent analysis that we did in the last 6 months. You can see each of the breed plan traits here. And for animals that started with a 30% EBV accuracy in conventional breed plan, when we didn't have genomics included, what we've done is we've looked at, well, let's put genomics in. What is the average increase that we see? So you can see here the gestation length, animals that had an EBV accuracy of 30% prior, when we included the genomic information, that lifted to an average EBV accuracy of 53%, which is a 23% increase in EBV accuracy. And you can see variable lifts for different traits. Retail beef yield and intramuscular fat, for example, on average have gone from 30% to 38%, so only an 8% rise, whereas 200-day weight has gone from 30 to 54, which is a 24% rise in accuracy. That's one breed. If we look at a second breed, we can see that for some traits like retail beef yield, we see the same thing. They went from 30% accuracy to 38% accuracy, so a lift of 8%. Whereas if we look at 200-day weight, this first breed lifted from 30 to 54. This second breed has lifted from 30 to 67. So we don't see what you observe in one breed is not necessarily what you'll observe in the other. It is really driven by the reference population that we have for that breed, for that trait. And so that's why we are continuing to encourage everyone to performance record as well as genotype animals. We really need to grow those reference populations and have them not just representing all of the different lines within the breed, but also representing having animals that are genotyped as well as got performance data for a range of economically important traits. And it's worth pointing out that these two breeds that we looked at, one is a Bos Taurus breed, one is a Bos Indicus breed. One has been using single step for a number of years. One is about 2 years into having a single step evaluation. So these increases aren't static. As we get more and more information into the reference population, we get more animals with genotypes, more animals with performance data. I expect the impact of genomics on EBV accuracy to continue to push up higher and higher for animals that started with a low EBV accuracy. It's then worth, I think, talking about, well, we've seen that we get we can get EBVs for animals that didn't have them prior. We've seen that we can get increases in EBV accuracy. But really the reason we're doing that is to see whether or not we get an increased rate of genetic progress in different breeds. And so what we've got here is 3 different breeds. This is breed A. We introduced single step for these 3 breeds back in 2017. And you can see for the 6 or 7 years prior, the index value increased by 24 Australian dollars. which is approximately $3.50 per year. Whereas in years after we introduced single step, you can see that it's increased by $39 with an average increase of $5.61 per year. So we're seeing the rate of genetic improvement tick up, and you can see that that hasn't happened straight away. It really is ticking up in recent years. The same for breed B. Prior to the introduction of single step, index value over this time period from 2011 to 2017 increased by $10.80, which is $1.50 per year. From 2017 to 2023, it's increased by $54, which is $7.79 per year. And again, we can see that uptick in recent years. And for breed C, much the same pattern from 2011 to 2017, index value increased by $7.30, which is an average of $1.04 per year. Whereas in the 7 years after we introduced our single step breed plan, it's $17, and which is $2.44 per year. So we're certainly seeing evidence of an increased rate of genetic improvement within these breeds. This is using their selection index, and certainly not seeing it as soon as single step is implemented. You can see there has really been some heavy upticks in recent calendar years, which is encouraging. And it really has mirrored the increased rate of genotyping. We have seen beef breeders embracing genotyping since the introduction of single step. And you can see for each of those three breeds, we're getting more and more animals genotyped each year. So that's been really encouraging to see producers start to genotype their animals. Anecdotally, we've seen them move from genotyping their sires to also genotyping the younger animals, so saleables, and also starting to genotype the female breeding herd. Have we had any questions come in?
Rina, we have one saying that she took DNA samples when the calves were just a month old. Is it worth redoing?
Also tail hairs. I mean, if it's worked successfully for you and the lab has got DNA, no. I have certainly heard anecdotally from people that they've taken them on younger animals, and it's worked. I guess the main thing is that the lab won't guarantee that it will work on animals that are younger than younger than 60 days of age. So I would say if it's worked, you're okay, but just be aware that the labs won't guarantee it, and you may not get DNA in each case. Was that the only question we have, Azan?
Yes.
Yeah.
Up until now, yes.
All right. So I just wanted to finish up with some considerations for beef breeders in the genomics era. So I wanted to talk about, should you keep collecting performance data as we go forward, and which animals should you consider for genotyping? So I think one of the biggest misconceptions is that many beef producers believe that they can stop performance reporting once genomics or single step breed plan is made available for their breed. Mike Coffey said this a while ago. In the era of genomics, phenotype is king. And I hope I've showed you that a little as well when I've talked about how we need those good quality reference populations to help drive the lift in EBV accuracy that we see. So single step works best when our current population is closely related to the reference population. So we need that reference population to be updated over time, and we need animals with phenotypes or performance data for a range of economically important traits, as well as genotypes or DNA going forward. As we've seen, genomics will likely give EBV accuracy a boost. We saw that animals that started with low EBV accuracy, when we added the DNA information in, we saw a lift in their EBV accuracy, but it's not likely to move animals into the high EBV accuracy territory. So consider DNA testing to really be an additional source of information rather than the only source of information that you collect. And any animal that you've got that has both performance data and genotypes will become part of the reference population. And for you as a breeder, that's useful because your animals are then closely related to the reference population, and that assists with the calculation of their EBVs. In terms of which animals you should genotype, it really depends on what you want your genotype results for. So single-step pre-plan, parent verification, are you trying to manage genetic conditions or qualitative traits in your herd? Usually people will be doing a combination of the above. In Australia, as I said, we've seen an update, an increase in the number of animals being genotyped since we made single-step pre-plan available for different breeds. We saw that earlier. And anecdotally, we're seeing producers moving from just doing the size and or saleables to genotyping the female breeder herd. I am certainly aware that there's a number of breeders who have been doing their female cohort each year and have got to the point where their whole breeder herd is genotyped. So we know that the cost of genotyping the entire female herd in one hit can be really prohibitive. One way to build up the number of females you have genotyped in your herd is to do your heifers each year. I would recommend doing the ones that you're definitely going to keep because they will go on to be the dams in your herd into the future. The maybes, if you would like to use that genomic information to go into the calculation of their EBVs and then make a decision based on the genetic merit of those animals, the maybes are a consideration to genotype. I don't think anyone is going to suggest that you genotype your cull females. They're not going to stay in the herd. If they're obvious culls, there's not a lot of point in genotyping them because you're culling them on structure, for example. It doesn't really matter what their EBVs are. They're not going to stay in the herd no matter what their genomic what the genomic information does to their EBVs. So in terms of how you can build those females up over time, if in the first year you do your 1-year-old heifers, when you head into year 2, you would do your 1-year-old heifers again for that year, and your 2-year-old heifers would already have been genotyped the previous year, and so on and so forth. So you see in year 3, you do 1-year-old heifers, and then the 2-year-old heifers and the 3-year-old cows are already done from year 1 and year 2, and so on and so forth until you reach a point where all of the animals in your herd are genotyped. So if you are interested in doing your females, but it's not an affordable situation for you to do them all in one hit, I would recommend doing your replacement heifers each year as a starting point. If anyone would like any further information, we do have a technical note on the case for genotyping females, which is available via the BreedPlan website. So just to finish up, we've seen in recent years the ability to take a DNA sample, send it to a commercial genotyping lab, and that allows you the opportunity to use DNA technology for parent verification, to manage genetic conditions, to manage qualitative traits, or for use in genetic evaluation, or a combination. We've seen that single-step pre-plan is delivering benefits for beef producers. We see more animals with EBVs available. We see animals with more accurate EBVs available, and we see that has helped to increase the rate of genetic improvement within some of those case study breeds. But as I said, if you remember one thing from tonight, please let it be this: performance recording or collecting phenotypes is still vital as we move forward in the genomics era. Thank you.